And this is going to be Chapter 3, Cell Structure and Function, and this is going to be Part 2. And in this section we're going to be talking about the cytoplasm and the cytosol and the organelles. These are going to be the fluid inside the cell and the actual little parts that make up the cell's environment or allow it to do what its job is or its intended function. 3.1, the cytoplasm. Cytoplasm is a general term for the material inside the cell from the cell membrane to the nucleus containing cytosol and organelles. So whenever we make the term cytoplasm, this is all the fluid inside of there and all the little special parts. And 3.2, the cytosol. Cytosol, intercellular fluid which contains dissolved nutrients, ions, soluble and insoluble proteins, and waste products. It differs from extracellular fluid in the following ways. Higher concentration of potassium and lower sodium. Remember whenever we talked about this before? Potassium was higher on the inside of the cell and sodium was higher on the outside of the cell. Cytosol contains a high concentration of dissolved proteins, many of them enzymes, so it can get enzymatic products out. Pardon me. It usually contains a small amount of carbohydrates and a large reserve of amino acids and lipids. Also contains insoluble material known as inclusions, glycogen granules, and lipid droplets. Next section we're going to talk about organelles. Organelles are internal structures that perform specific functions essential to the normal cell structure. Membrane enclosed organelles are going to be the nucleus, which is going to be the center of the cell, and then the mitochondria, which is going to be the powerhouse. Endoplasmic reticulum, and we have two types here, smooth and rough. Golgi apparatus, we're going to see exocytosis, that term that we learned earlier in the previous slide set. Lysomes, which pretty much break down the cell, and then peroxisomes, which oxidize things. Non-membranous organelles, cytoskeleton, this is structure. Microvilli, little finger-like projections on the surface of the cell. Centrioles, cilia, flagella. Centrioles are going to be on the interior, allow for supportive structure. Cilia and flagella are either going to be little hair finger-like projections, or flagella is a tail to allow for movement. The ribosomes, which create proteins, and proteasomes, which will break down proteins or destroy denatured proteins and things like that. And we're going to talk about all of those in this section. Cytoskeleton. Cytoskeleton is an internal protein framework of various thread-like filaments and hollow tubes that gives the cytoplasm strength and flexibility. Microfilaments, which are the thinnest strands of the cytoskeleton, and they're made of actin. Thick filaments in muscle cells made of protein and produce contraction. So whenever we talk about muscle cells in the following chapters, we're going to see something that has a thick filament and a thin filament. And pretty much what these two do inside of a muscle cell is whenever a muscle contracts, the thin filaments fit inside of the thick filament. And the myosin head and the actin kind of bring the two filaments together. Intermediate filaments, medium-sized filaments stabilize its position with respect to surrounding cells, and then microtubules, hollow tubes built from globular proteins, give cell strength and rigidity anchors organelles. So all those little organelles that are floating around freeform in the actual intercellular or the cytoplasm, all of those are got to be anchored, and they're anchored by microtubules. And here are some structures. Let's take a look at this. These are microvilli, these little finger-like projections. You'll notice that they have quite a bit of structural proteins inside of them. These right here, the purple things, are the intermediate filaments, and this is figure 1315. These are secretory vessels, whether it be through exocytosis or this is a product of endocytosis. Microtubules, which allow other structures to kind of anchor themselves. Another microtubule, and here we have a mitochondria. And then we have microfilaments, which are here, provide structure and form. And the microvilli, if this was cilia that were in the lungs, this would provide surface area for oxygen and CO2 to come out of. Centrioles, cilia, and flagella. Centriole is a cylindrical structure composed of short microtubules. Cilia, relative long, slender extensions of the cell membranes. Movement requires energy. If small intestines or something that are ciliated, the lungs ciliated for surface area. Small intestine, same reason, surface area. They also have a wave-like fashion from the muscular structures underneath them that will allow us to move food and substances into the lower intestine. Flagella. Organelles resemble cilia but larger. Move cell through fluid. And an example of something that has a flagella on it is a sperm cell. It's a kind of a tail that assisted in moving. 3.7 ribosomes. Organelles that manufacture proteins. Each consists of a small and large subunit composed of ribosomal or RNA and protein. Two major types. We have free ribosomes. Ribosomes scattered through the cytoplasm, and the protein they manufacture enters the cytoplasm. And then we have fixed ribosomes. Attached to the endoplasmic reticulum, a membranous organelle. Proteins manufactured by fixed ribosomes enter the endoplasmic reticulum, where they are packaged for transport. And these would be proteins that we are sending out to other locations. Proteasomes, hollow cylindrical organelles that contain an assortment of protein-breaking enzymes, removing and recycling denatured proteins. And if a protein's bad or doesn't make it past the checklist, genetical checklist, what these proteasomes do, they'll just break down the protein and reutilize it for its parts. Endoplasmic reticulum. The ER is a network of intracellular membranes connected to the membranous nuclear envelope that surrounds the nucleus. Has four functions, and if you'll pay attention to these are going to be important. The ER synthesizes proteins, which means it makes them. It stores proteins. It transports proteins, and it provides detoxification. If we have anything, drugs or anything like that, that are going to be absorbed by proteins or require a protein bath for biotransformation, this is what's going to do it. So synthesis. Special regions manufacture proteins, carbohydrates, and lipids. And this is the endoplasmic reticulum. Storage. The ER can store synthesized molecules or material absorbed from the cytosol, and this is going to be inside the cell, without affecting other operations. Transport. Materials can be moved from place to place in the endoplasmic reticulum. Detoxification. Drugs or toxins can be absorbed by the ER and neutralized by enzymes within it. Four functions of the endoplasmic reticulum. Smooth endoplasmic reticulum. Lipids and carbohydrates are produced in here. Smooth endoplasmic reticulum functions. Synthesis of phospholipids and cholesterol is going to be very important in the making and the manufacture and the keep up of the cellular environment. The synthesis of steroids and hormones. So it's actually going to make steroids and hormones, and these will interact with multiple organ systems throughout the body. Synthesis and storage of glycerides, and synthesis and storage of glycogen. So glycogen is a polysaccharide, which means it's many chains or many molecules of sugar. Rough endoplasmic reticulum. Combination workshop and shipping depot is pretty much the Barney answer on this. Fixed ribosomes release newly synthesized proteins. So if it's going to make a protein, this is where it's going to release it from. Ship and delivery proteins to the Golgi apparatus, which if it requires packaging and sent outside the cellular environment, then the Golgi apparatus is what's going to do that. And that's going to be coming up in just a second here. So we have two types of endoplasmic reticulum. We have smooth and we have rough. The rough endoplasmic reticulum essentially has ribosomes in it, and that's what gives it these little speckled appearance here. The smooth endoplasmic reticulum does not have ribosomes in it. Ribosomes are going to assist ribosomal RNA. Ribosomal nucleic acid, is what is actually going to help us synthesize DNA. Golgi apparatus consists of a set of 5 to 6 flattened membranous discs. Major function of the Golgi apparatus: modification and packaging of secretions, hormones, and enzymes, renewal or modification of the cell membrane, packaging of special enzymes used in the cytosol or inside the cell. Apparatus has 3 types of vesicles, and this thing is going to either package them, apply for renewal, or make stuff for the cellular environment, cell wall, and things like that. Secretory vessels, secretions that will be discharged from the cell. So this is stuff that we've made, packaged up, and now we're exporting to other cellular environments. And then membrane renewal vesicles fuse with the surface of the cell and add new lipids and protein to the cell membrane. Golgi apparatus. And this is that exocytosis and endocytosis. This is what the Golgi apparatus kind of runs here. I want to take a look over here. This is exocytosis actually occurring, where it's expelling cellular material outside. This is the cell wall here. Vesicles incorporate into the cell membrane, and this is membrane renewal. So these things will travel up and then attach itself and add to the cell membrane. And these are secretory vessels. So if we had some hormones or steroids that we needed to put into the system, this would just be brought to the surface and then expelled into the extracellular environment. And that's pretty much the 2 different types. Lysosomes. Vesicles filled with digestive enzymes perform cleanup and recycling functions within the cell. Defense against disease. Lysosomes fuse with organic debris for initial digestion, and their pretty much job in life is to destroy the cells. So they're kind of like a self-destruct mechanism inside the cell. Peroxisomes. Smaller than lysosomes and carry a different group of enzymes. Still break down stuff though. Absorb and break down fatty acids. They do generate, which is a nasty byproduct, hydrogen peroxide, which is a potent, dangerous free radical. If we have a ton of oxygen in the extracellular environment, that's kind of what ages us, is oxygen and the way that we use the mitochondria to generate energy. And these peroxisomes are pretty much the most abundant in the liver. And that's because the liver's job in life is to pretty much oxidize substances, and it has about 200 different functions. Whenever we get to it, we'll pin down a few of them on it. Mitochondria produces energy for the cell, and this is kind of like the powerhouse of the cell. Most chemical reactions that release energy occur in the mitochondria. Energy is stored in the form of ATP, which stands for adenosine triphosphate. Cells break down ATP, adenosine triphosphate, to ADP, adenosine diphosphate. And again, we're going to see the ATP to ADP. We're just essentially using the phosphate as an energy supply. 4 general steps to ATP. And step 1 is going to be glycolysis, which is broken into pyruvic acid. And if we'll go back to this, the breakdown of sugar, glycolysis, we pretty much turn this into pyruvic acid because the glucose itself is not utilized well within the mitochondria, so it changes into another form. The pyruvic acid is absorbed into the mitochondria. If oxygen is present, we'll break down into carbon dioxide and water. And water, which is byproducts of cellular metabolism, CO2 and water. And enzymatically, which means that it's going to take part A and part B and turn it into part D. So it's enzymatically going to convert adenosine diphosphate to adenosine triphosphate. This term will be utilized again and again and again throughout this entire course. Aerobic metabolism, or cellular respiration, produces approximately 95% of what the cells need in its metabolism or the production of ATP to survive. 95% aerobic metabolism. And this is a picture of the mitochondria. Glucose to pyruvic acid through the mitochondria, and through its enzymatic action we get ADP changed into ATP, and we're going to continuously burn and restack that phosphate molecule, and we'll actually make some extra too, as we see in the muscle chapter. The nucleus, chromosome structure, genetic code, and then protein synthesis in this section. Nucleus, largest structure in the cell, control center for cellular operations, all information needed to synthesize more than 400,000 different proteins in the human body. Nuclear envelope, double membrane that surrounds the nucleus and separates its fluid contents, contains ions, enzyme, ribosomal nucleic acid, deoxyribonucleic acid, nucleotides, proteins, small amounts of RNA and DNA, nuclear pores, which is another term, ports large enough to permit ion movement and small enough to regulate the transport of proteins and RNAs. Nucleoli, organelles that synthesize ribosomal RNA, which is our RNA, ribosomal RNA, the site of protein synthesis in all living cells. So those ribosomes on the endoplasmic reticulum make proteins. That's all they do in life. Figure 3.19 in your book, which is the nucleus. This is the picture rendering here, and then this is the actual blown up electron microscope version. So we have the nucleolus here, and this is it in the actual cell, and then we have the chromatin, which is nuclear material. There's a picture right there. Nuclear envelope, which is the surface on the outside, and seen here. And then we have nuclear pores. The nuclear pores, little tiny openings and holes that allow ions and things like that, but will not allow protein structures or codons to come out of the nuclear envelope. Chromosome structure. Chromosomes, DNA in the nucleus that stores instructions for protein synthesis. So this is kind of like the roadmap to how the body works. Contains 23 pairs of chromosomes, 1 pair from the mother and 1 pair from the father. So you have 46 individual chromosomes. You get 23 from mom and 23 from dad. A chromatin, DNA loosely coiled that forms a tangle of fine filament. And we're going to see these kind of all intertwined here. So this is the nucleus, and it's preparing for cellular division. So it's going to separate off, it's going to make 2 cells. And these are the daughter chromosomes, and this coiled region here is pretty much, these are histones. All of this stuff here is circling around. It looks like a cord that's wrapped around it is actually the DNA helix. So all of this is your actual nuclear material. This is the instructions for what this cell was supposed to do in life. The genetic code. Each protein contains a unique sequence of amino acids. Genetic code, chemical language of the cells. And a gene is a functional unit of hereditary. Each gene consists of all of the triplets needed to produce a specific protein. And then here's where we come into the word codons. Triplet code, sequence of 3 nitrogen bases needed to produce a specific protein. So whenever we get DNA replication occurring, those 3, like AUG, and this would be after it, the DNA was kind of unzipped by the DNA polymerase, and it transcribed it into a 3 protein subunit, and that 3 protein subunit is called a codon. Clinical note, and at this point, DNA fingerprinting, and pretty much everyone would know this if you've ever watched an episode of CNI. Every cell contains 46 single chromosomes. The DNA material is kind of unique. It's a chemical fingerprint, so you won't find 2 people that are identical unless they are absolutely identical twins. And we'll talk about that later whenever we get into hereditary, about chapter 19. So DNA fingerprints are pretty much unique in that you can identify one person from the next. Protein synthesis. Protein synthesis, or the production of protein, divided into 2 parts. We get transcription and translation. Transcription is the production of RNA from a single strand of DNA. Translation is the assembly of protein by the ribosomes using the information carried by the RNA. Transcription takes place in the nucleus. Translation occurs in the cytoplasm. So again, an overall view so we can kind of see this. We got the cell, we got the nucleus here. The transcription and the unzipping of this DNA occurs inside the nucleus. So you get these 3 protein codons that are coming off of this. Those then are transferred to the rhizomal RNA, and what they will do is they will assemble a protein sequence for you. So this is translated, or translation occurs in the cytoplasm. So in the ER, where you have rhizomal RNA in the ribosomes, these 3 codons then are translated from what the messenger RNA has brought out of the nucleus. mRNA, we have tRNA here that will transfer to the ribosome, and then we get DNA production. Transcription occurs in the nucleus, makes a strand of messenger RNA, utilizes adenine, guanine, cytosine, and uracil, A, G, C, and U. Messenger RNA, a single strand of RNA, is formed in the nucleus. Transcribing or copying is achieved. Transcription, the process of messenger RNA formation, and a codon is a sequence of 3 nitrogen bases along an mRNA strand, and that is considered a codon. And again here, remember whenever we're going from DNA to DNA, these 2, this one here, this uracil, is only in the RNA. The DNA has thymine, which is, the T is right here. But as this DNA is unzipped, the RNA replaces A. In the DNA it would be A to T, but in the messenger RNA it is A to U. Now once this exits out of the cell and this goes to the rhizomal reproduction, the mRNA strand then is translated. This U will be translated to a T on the other side as it's translated. So again, DNA polymerase unzips the DNA. You get 3 codon sets, 3 protein sets that are considered codons. The mRNA comes into here and it makes essentially a chemical carbon copy of this codon. That then is pulled outside of the nucleus. Once it is translated, it will make an exact copy of that 3 protein sequence. Translation. Translation, synthesis of a protein using the information provided by the sequence of codons along the messenger RNA or the mRNA. transfer RNA, or tRNA, a delivery device from the nucleus to the ribosome to assemble a protein mirror image of RNA, more than 20 different types. And then an anticodon. If a codon is a 3-protein set, an anticodon is a complementary triplet of nitrogenous bases that will bind to a specific codon of mRNA. So in the rhizomal area of the ER, you have all these proteins. So as you got this protein or this messenger protein coming in to be translated, those proteins will attach to it. And those 3 proteins that attach to it are enzymatically put together, and then that makes a codon. Well, that is going to be a replicant of what is inside the nucleus. It's just a long way of doing it. So translation, part 1 here. In the nucleus, you get messenger RNA, which it comes out. The mRNA binds to a small ribosomal subunit and is joined to the start codon by the first translation RNA, which carries the amino acid methionine. Binding occurs between complementary base pairs of the codon and anticodon. So, ribosomal subunit, if you have the anticodon, the start codon of AUG is going to be UAC, and so on. CCG, GGC, 3 codon pairs, dot, 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 and it goes on making these long protein chains again. The second tRNA arrives at the adjacent binding site of the ribosome. The anticodon of the second tRNA binds to the next mRNA codon, and so on and so on and so on. We actually, in the previous slide, kind of jumped ahead to about this step here. So the first amino acid is attached from its tRNA and is joined to the second amino acid. So again, AUG, U, A, C, and this thing is spitting out a protein chain, essentially. Once this protein chain is completed, it's kind of free-floated, and again can be, if it needs to be zipped up, or they have a continuous running of DNA here, DNA polymerase will zip this thing back up to where it's a DNA string. Clinical technology, a recombinant DNA technology. Now, they do this in several arenas, and what this is they essentially tell a bacteria to produce human insulin by giving it specific codons. So what this does is this recombines the DNA production of, let's say, bacteria, so that this bacteria now will produce human insulin. Other examples of recombinant DNA technology are going to be hepatitis B, and then clot-busting agents. Tissue plasminogen activator is the one that it's listed here, TPA. And this is done, we pretty much early use something called strep. Everyone's heard of strep throat. Streptokinase is a type of tissue plasminogen activator, or TPA. So what this does is this was made from the strep bacteria, and it reconstitutes DNA or rhizomal units to where it is producing tissue plasminogen activator, which, if put in the body, will activate the plasminogen in the tissues. This essentially tells the clot to dissolve itself. Cell life cycle, interphase, mitosis, cytokinesis, cellular division, and cancer. So cell life cycle, cell division occurs, and from fertilization, or the formation of a zygote, to the mature organism, the body goes from 1 cell to 75 trillion cells. Mitosis is nuclear division of the cell, or the separation of the cell. Somatic cells, diploid, is any biological cell forming the body of an organism. Meiosis is a special type of cell division necessary for sexual reproduction. Interphase is the phase of a cell cycle in which the cell spends the majority of its time and performs the majority of its purposes, including preparation for cell division. Interphase. And all of this here is interphase. This part here is where we get mitosis, or actually the cell is going to divide and make one of its offspring or daughter cell. So let me get this erased here really quick. So 8 or more hours we get in interphase, and what it's doing is preparing, and this part of it here, 6 to 8 hours, DNA replication, synthesis of histones, and this is before the nucleus divides. 2 to 5 hours, and this is protein synthesis, or the production of the DNA for the daughter cell. And then once we got all the parts made, what we get is the starting of cellular division. And then this right here where it says cytokinesis, this would be the production or the breaking apart or the actual separation of parent cell from daughter cell. Interphase, the phase of a cell cycle in which the cell spends the majority of its time and performs the majority of its purposes, including preparation for cell division. An apoptosis is going to be a term for cell death. DNA replication occurs in interphase, which is copying of the genetic information in the nucleus, so that one set of chromosomes can be given to each of the two cells produced. DNA polymerase, an enzyme that helps catalyze in the polymerization of deoxyribonucleotides, which means that it allows an environment to make DNA. Polymerase reads the intact DNA strand as a template and uses it to synthesize a new strand. And this right here is going to be an illustration or a picture. This is figure 2.24. And this is the DNA polymerase, and this is what I was referring to in the other slides as the zipper. If you'll notice it is the DNA, this DNA polymerase hits the DNA, it unzips it. This allows for those codons to be made and the new production of DNA. Mitosis. Mitosis is a process that separates and encloses the duplicated chromosomes of the original cell into identical nuclei. Cytokinesis, cytoplasmic division of daughter cells, usually begins in late anaphase, and that's this part down here, the second to the last phase, and then is pretty much completed in telophase, which means instead of 1, I now have 2. Alright, so this is mitosis. And I'm going to kind of talk you through this. There's some reading as far as prophase section, telophase section, anaphase section, not specifically in that order. So, nucleus, this is interphase. This is where it spent most of its life. It's now making parts so that it can become 2 cells. Mitosis begins. Centrioles, you essentially get 2 pair. And they'll pretty much make polar ends. Sometimes this can be seen under the microscope. This is early prophase. By late prophase, we have polar ends, and the chromosomes now are forming to where eventually, whenever this thing starts to separate, they'll form right along this line. In metaphase, how we identify it is the chromosomes have a metaphase plate, which they line up right in the middle. In anaphase, we see separation of the chromosomes. In telophase, and anaphase and telophase is where we get cytokinesis occurring. Definitely by late telophase, we'll get 2 cells. But the thing about telophase is we start to see the cellular walls kind of get themselves is called a cleavage furrow. So what's going to happen here is these cells are going to say, okay, I'm cell number 1 and I am cell number 2. And they'll separate right here in the center. And then whenever cytokinesis occurs, or the production of a daughter cell, that's what we get. We started off with 1 cell, but now we have 2. Cellular division. Mitosis. Prophase begins with the chromosomes coiled so tight they can be seen by a light microscope. Now there are 2 copies of each chromosome. A chromatid is one of 2 identical copies of DNA making up a duplicate chromosome, which are joined at the centromeres. For the process of the cell division, mitosis or meiosis, a centromere is a region of DNA typically found near the middle of a chromosome where 2 identical sister chromatids come closest in contact with one another. Spindle fibers are the structure that separates the chromosome into daughter cells during cellular division. This is metaphase. Begins as the chromatids move into a narrow central zone called a metaphase plate, and this is where you have polar ends, the spindles, and the chromosomes were right there in the middle. Metaphasic plate. Metaphase ends when all of the chromatids are aligned in the plate of the metaphase plate. Anaphase begins when the centromere of each chromatid pair splits and the chromatids separate. The 2 daughter chromosomes that result are now pulled towards opposite ends. So in the picture you had spindle fibers, so these things kind of separated off and they're pulling towards opposite ends of the cell. Telophase. The cell prepares to return to interphase. The nuclear membrane forms, the nucleoli enlarge, and the chromosomes gradually uncoil. Once the chromosomes have relaxed and the fine filaments of chromatid become visible, nucleoli reappear, and the nuclei resemble those of interphase cells. Again, nucleus. I spent all my life ready to make a daughter cell. Early prophase, 2 centriole pairs. These are going to make our polar ends. Late prophase, we have polar ends, and we start assembling the chromosomes here in the middle. When metaphase occurs, we have a metaphasic plate. This is the reason this is called metaphase. It's beginning to separate. In anaphase, we have daughter chromosomes. Essentially 2 pair, but they still aren't separated out in the middle yet. In telophase, we start to see a cleavage occurring, or the separation of the 2 cells. And whenever cytokinesis occurs, we have 1 cell here and another cell here. So it started with 1 and now we have 2 cells. Cytokinesis is the process in which the cytoplasm of a single eukaryotic cell is divided to form 2 daughter cells. It is the division of the cytoplasm. Cytokinesis. Cell division in cancer. So, tumor, neoplasm, a mass or swelling produced by abnormal cell growth or division. Again, one of the reasons that this can occur is because we get 3 codons here, or 3 protein nitrogenous bases that started off in the nucleus, got translated, got spit out of the nucleus, got transcripted in the nucleus, got translated outside the nucleus, and then got produced into another one, into another complete protein chain, or zipped up as DNA. Now, the problem with this is do you think after all those millions upon millions of encodings that we can get a run of mutation or a run of protein chains that may not code correctly? This is why we get these guys right here, and tumors. Now, and there's different types of tumors, since we're talking about these. Benign tumors, pretty much no harm, no foul. Abnormal cells, and they're confined within a connective tissue capsule. So, whenever we're looking at these benign tumors, they have their own little capsule, they're their own little organism, and they're keeping to themselves. A malignant tumor. Tumor that cells no longer respond to normal control mechanisms, do not remain in the connected capsule. So these things went from a benign, encapsulated, funky cell that shouldn't be there, into something that is aggressive, it's moving into surrounding tissues, and so on. Invasion, the spreading process into surrounding tissues. Metastasis, or metastatic, traveling to distant tissues and organs that produce secondary tumors. So this may have started in your head, or it may have started in your foot, and now it's in your hand, or now it's in your stomach, or now it's in your leg, or now it's in your brain. So metastatic cancer means that it moves into the surrounding tissue that are not just adjacent to it. It could have started off in your toe and now moved into your brain. And cancer, illness that results from malignant cells, or cells that, the problem is every one of these cells have the ability, and they know what their job role is. So whenever you get cancer cells into, let's say, an organ that has a specific function, that's going to be problematic. The reason it's going to be problematic is because the cancer cells may not function like the surrounding tissue. Example of this, if you had a tumor that was growing in your heart needs to contract rhythmically with the surrounding tissue simultaneously. If this is not occurring because you have a section of real estate in there that is essentially a cancer tumor, it may not contract the way it should. You're going to lose what is called ejection fraction, or blood ejecting from those ventricles. Well, that's going to be problematic. So illness that results from malignant cells is cancer. Cell diversity and differentiation. So differentiation is the process by which a less specialized cell becomes more specialized cell type. An example of this is going to be liver cells, heart cells, lung cells. They all come from the same cells. So when a cell starts off, until it knows what it wants to do in life, it's a blank. I don't know what I want to be. It isn't until the DNA comes out and it says, well, I wanted you to be a heart cell. So until it knows what its end result is, it is less specialized. It isn't until it becomes intact with its environment, or it knows what its job in life is. An example of this would be an embryo. The cells all start off essentially the same. As they become more specialized, some of the cells will make lungs, some of the cells will make heart tissue, some of the cells will make liver tissue. But they all started off from a blank cell called a stem cell. If you have questions over this chapter, this is chapter 3, part 2, cell structure and function, feel free to call me, 219-7613 or smithr@emsa.net. Thank you very much.